Good afternoon, students. Welcome back to biology classes, and already we have started with mechanism of replications. Okay. So in mechanism of replications, how DNA is going to make copy in prokaryotes, eukaryotes, and all. So first step is initiation. Okay, first step is an initiation of the replication. So, how the initiation is going to be started. So, as we have told that in any DNA, if you take, they have a particular site which is called as ora. Ora is nothing but where the replication will start. Okay, the origin, start, initiate. So it is that site initiation of replication start at the origin, and it is the process of replication is by semi-conservative method, semi-discontinuous, and bidirectional. These three terms you have to remember. Okay, so why it is called bidirectional? Think this is a replication is taking place in the nucleus from the upper and towards the here origin, and here terminal is taking place. Okay, origin as well as the terminus. So both the side it is taking place, this side, and again the replication also starts from this side. So both the side it is taking place, hence the replication is bidirectional. And we have also seen that the DNA replication if we take. So the DNA replication, during the DNA replication, so here the one thing is, so two DNA new formation will take place. In that, one will be sharing with the old strand, act as a template, parental strand, and one see here, this is the parental strand. Both the polynucleotide chain act as a template. Now the two DNAs are formed. In the two DNA copies, one strand will be exactly similar like a parental strand, and another strand is totally new. Okay, from where it is originated? Here in eukaryotes, the DNA is twisted. In prokaryote, only one ori will be present, but here many ori can be seen in the eukaryotic DNA. So initiation start. So hence they are called conservative method. Half parental character, half parental DNA copy, half new DNA will be synthesized. Okay, so they are called semi-conservative. Okay, here if you draw the diagram of the formation of DNA replication, here unwinding is taking place, and so next and all you are going to study. Why it is called the discontinuous? So during the formation, one is continuous, another one will be in the form of fragments. So it is semi-discontinuous. Okay, during the replication time, so the parental strand complementary to the inner new strand will be formed. That inner new strand which is formed, one strand, first strand is continuous. Second strand on the second parental template, it will be discontinued. Hence it is called as one is continued, the other one is discontinued. So it is called semi-discontinuous. Coming to the mechanism, so the initiation has started. Second step is activation. Activation, what way they have by using an enzyme. By using the enzyme, that is DAMP, deoxyadenosine monophosphate, okay, deoxyguanosine monophosphate, deoxycytosine monophosphate, deoxythymosin monophosphate. These should be converted into, it should not be in monophosphate, that should be converted into triphosphate in the presence of enzyme, that is phosphorylase enzyme. So by addition of phosphorylation, monophosphate will convert into triphosphate, like deoxy adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, and thymine triphosphate. Okay, so these they are going to convert. When they convert into triphosphate, they will become activated. Okay, this is second step. First step, the initiation will be started. Second step is what? They are going to convert themselves into trioxide. They are going to convert into. Then, after that, this will play a major role. Triphosphate helps in forming the H bond. Next, third, what is this? Unwinding. Unwinding, we have drawn the last class the diagram. By adding helicase enzyme, it will help the DNA to unwind. You can see the unwinding is taking place. Okay, so unwinding of the DNA helix with the help of enzyme helicase, and also here other proteins are also present on this. Here helicase enzyme is present. This helicase enzyme is responsible for unwinding of the DNA, and also after unwinding, the DNA template should not coil. So we are using single SSBP, that is single strand binding protein. Okay, so this single strand binding protein, they will attach to that strand and coiling will be avoided. Next, after the formation of this activation and all, third, next. Unbinding has taken place. Next, for synthesis, still this has not been formed. Okay, for synthesis of DNA, they need RNA primer. Okay, we are at the fourth step. RNA primer is already, I explained this. This is a small, where it is moving, a small having 3 5 prime. Especially this will be formed. Here 5 dash 3 prime. Okay, here 3 prime and 5 prime. Always RNA will be formed towards 5 prime to 3 prime direction. How it is going to move? From 5 prime to 3 prime direction and forms the template. This is a formation of RNA primer. Okay, this is a what movement? What you are seeing? This is synthesized. And now 5 dash to 3 dash direction. Here RNA is formed. Here one RNA is formed. Here RNA primer is formed. So again RNA primer is synthesized from the DNA only. Okay, it is synthesized from the DNA itself. But this RNA primer is responsible for the formation of the new strand. Okay, so RNA primer is synthesized. Once the template is synthesized, that RNA primer should be removed by an enzymes. Okay, topoisomerase is there, DNA polymerase enzymes we have. Okay, so we have to look. So first RNA primer, formation of RNA primer is also important. Next. After formation of RNA primer, so new strand with the help of enzyme kinase, and RNA primer is synthesized in the presence of RNA polymerase. How these are synthesized? Because of the RNA kinase enzyme is responsible for the formation of RNA. So next step. After that, RNA polymerase, in prokaryotes we have different types that we have already discussed. No need of discussing here. Next, let us go for base pairing. From here we need to explain, right? Now, base pairing. So base pairing is nothing but one. The two strands which has replicated, they have to form the Replication fork formation. Think this is a DNA. The DNA is going to form like bubble-like structure and fork-like structure. This is a replication fork. Okay, the fork formation. Okay. So here what happens is deoxy triphosphate which was present, they are all converted into just write the reaction here. So, as we know that it was formed with deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, deoxythymine triphosphate. This is treated with so they have to again converted themselves into monophosphate in the presence of enzyme, pyrophosphatase enzyme. In the presence of pyrophosphatase enzyme. Okay. Now, earlier to convert monophosphate into triphosphate, we were using the enzyme. What type of enzyme we were using? Phosphatase enzymes. Here we are using pyrophosphatase enzyme, which will make triphosphate to convert into what? Monophosphate. Okay, to break the hydrogen bond. Okay, now this is treated with the they will convert into monophosphate. How they are going to again DAMP plus 2 inorganic phosphate is released. This is helpful for the breaking of the hydrogen bond. Breaks the hydrogen bond because in unwinding there is a what the hydrogen bond will be broken when with the help of energy. Okay, here this is going to apply here where this will help to what breaks of the hydrogen bond, unwinding will take place. Okay, so this after forming the mono energy where hydrogen bond is formed. So this energy is released for the pyrophosphatase which will form deoxyribonucleotides. Base pairing means what? By adding pyrophosphatase enzyme. So here the nucleotides go on attaching with the and forms polynucleotide chain. It forms polynucleotide. They will go on adding to 3' to 5' direction. So they will go, nucleotides will go on and increase the length. So by adding base pair, by using enzyme, and so after that energy is released and base pairing will be utilized. Next, chain formation. So here in presence of magnesium, energy utilization is taking place, right? So here the DNA polymerase 3 adjacent nucleotide is nitrogen base or template DNA will be formed. Okay, think this is a base and they are joined and phosphoester di formed later. Okay, so that is phosphodiester by using energy all the DNA by DNA polymerase enzyme alpha, beta, gamma, delta, epsilon they are responsible here. Okay, next. After this, next one by using DNA strand will be established by using diester bond and get linked to the bond, linked to the DNA strands. Next, replication. So DNA template. Now the ones these are formed, RNA primer should be removed. With the help of DNA polymerase 1, DNA polymerase 1 removes RNA primer. Okay. The enzymes already we have discussed about the enzyme. Once the strand has created, RNA will disappear. RNA should remove by the enzyme DNA polymerase 1. After this, next, while forming this thing, you can see the RNA primer will give rise to the wherever the 5' to 3' direction, the RNA primer will form and the phosphodiester bond is created. So, which is formed first and also continuous. So, this is called as a leading strand. And the strand which is discontinuous is called as lagging strand. First form, this is later form. Now this here the lagging strand what you are seeing is they do not have continuous formation there. They don't have the continuous formation here, right? So each strand is formed from the each RNA primer. Okay, we have fragments. Each fragment is formed from RNA primer. We have to remove RNA primer by using DNA polymerase 1, and now these will form the Okazaki fragments. What are Okazaki fragments here? Okazaki fragments are those which are not, the DNA which are formed are not continuous. Okay. So elongation of the strand will be taking place here. Elongation of the Okazaki fragment should be elongated there. They have to form continuous now. Which is responsible now to join these fragments on the lagging strand? Now after joining it will become lagging strand, right? What enzyme we have to add? So on the second DNA, it is discontinuous as I told you, and small fragment deposit RNA primer is also present there. Okazaki fragment. Okazaki fragment is having 1000 to 2000 nucleotide. Okazaki fragments have 1000 to 2000 nucleotides in prokaryotes. Okay, 100 to 200 in nucleotides in eukaryotes. Just a minute. So, Okazaki fragment, they have the 1000 to 2000 in prokaryotes. Okay, and after that in the eukaryotes 100 to 200 nucleotides are present. Next. So after deposition of Okazaki, RNA primer is released by the gap. Okay, with the help of polymerase, new strands is formed there. So here we are using with the by the Hg Kurana. Okay. Hg Kurana has introduced the DNA helicase. Hg Kurana has introduced the what? That is DNA ligase enzyme was deposition about explanation was given by Kurana. Okay, which is discontinued. So now Okazaki fragment is released. DNA polymerase is treated there. DNA polymerase enzyme is added to the Okazaki fragment and form the continuous in this direction. Okay. This is going in this direction and this is forming in this direction. Okazaki fragment, by treating it with the DNA polymerase, will patch up, the fragments will be joined, which is called as lagging strand. Okay. Termination means end. Okay. Termination is nothing but what? Thus, the strand is while the other strand which is discontinued. Hence, DNA replication is semi-discontinuous because Okazaki fragment is there. So, fragments was in the form of patches. Hence, they are called Okazaki fragments. One is continuous, another one is discontinuous. So, it is also called semi-discontinuous also. Okay, and whatever the new strand is formed, they are moving in what direction? Okay, they are moving in the 5 to 3 direction and here 5 to 3 direction. Both the side, the replication is taking place and the new strand which is formed, it is complementary to each other and the energy which is formed, they form the hydrogen bond. Okay, energy which has been produced through inorganic phosphate. They will help us to form the hydrogen bond between the new and the old synthesized. And later, topoisomerase enzyme will help them to replicate and they will be separated. Okay, next. Proofreading, last. Proofreading of the DNA. Sometimes what happens, the DNA will be coding. Okay, DNA polymerase removes the wrong base and attaches. DNA polymerase 1 is the DNA polymerase 1. Especially, this will remove if any mismatching. For example, thymine and uracil. Okay, sometimes by mistake they have been coded. They have not recorded. Instead of thymine, they have been coded for uracil. In that case, rechecking. Okay, proofreading is nothing but what? Recheck if any mistakes are there. Again, they will recheck. The enzyme DNA polymerase 1 enzyme is helpful for what it is helpful? It is helping the DNA for checking whether it has been sometimes thymine and instead of thymine they will code for uracil. Instead of uracil, thymine will be coded there. So in that case, they will recheck once again if any mistakes are there during coding. A to T, C to G, they will be coding. So T will be coded for the A. Sometimes instead of thymine, uracil also will be coded there. So it is a proofreading. So polymerase 3 able to check the error and removes the wrong base. Polymerase 1 removes the wrong base also and attaches the correct base also. Okay. Now prokaryote whereas polymerase beta in eukaryotes debate will take place. 1 in 10,000, always remember 1 in 10,000. 1 in 10,000 mutation may take place, otherwise it will be normal. Every 10,000, one member, one person will be having this mutation, or else the DNA is. So by this we have completed the mechanism of replication. First class we have studied all the enzymes, and the second class we have seen the diagram how they are, and the third we have completed the steps. So you have to write all these steps. You have to explain all these steps of the what of the replication if they ask you. So after this we will get the strand. 8 steps you have to remember. Lagging strand, after this, after combining all this, we will get the diagram. So you can see the synthesis of the DNA, how it is moving. The DNA formation is moving from this side to this side. Okay. Here fragments are moving from the 5 prime to 3 prime, 5 prime to 3 prime. Especially 3 prime to 5 prime template DNA. This is the parental strand. One will be parental, another one will be what? Newly synthesized DNA. Okay. So what we have completed here, 8 different steps of the replications we have completed. RNA world we have completed. Okay, packaging also we have completed, and also this one. Griffith experiments we have completed, Hershey Chase experiment also we have completed. So next we will left with the gene, okay, genetic code and all we have to left. So please go through this what we have studied. Just I'll recall once again the mechanism of semi-conservative method. First thing is initiation, very important for 5 marks. Basic knowledge what we have to think is explain replication using semi-conservative method. Yes, why it is called semi-conservative method? The parental strand act as a template. One parental strand and another one will be synthesized. One parental and another one will be synthesized. So from one parental, two DNA are going to be formed. Two DNA copies are formed. One point. And these are formed as a complementary to the parental. If the 3 prime to 5 prime, the newly synthesized strand which is there, it will move from 5 prime to 3 prime, 5 prime to 3 prime. If we will come to know one strand, automatically the other strand will be what? We can make the copy of the that. As we have told that initiation, okay, initiation where it is going to take place. Okay, initiation of this is going to be taken place in the, especially where it is taking initiation of the codon, that is origin is taking place, both the side it will take place, hence it is bidirectional, semi-conservative, bidirectional, and these are discontinuous, semi-discontinuous, because one is forming continue, okay, another forming in the form of fragment which is formed later. So they are called leading strand which is continue and lagging strand, and always the 5 direction, 3 direction, RNA primer second. We'll see that. So these are mean complementary. The newly synthesized DNA is complementary, right? Next, activation. So it was present in triphosphate. They are all treated with the enzyme, that is phosphatase enzyme, and they will convert into monophosphate. And once they will convert into monophosphate, activation of the nucleotides will start. Okay, activation of the deoxyribonucleotides activation will take place. Third, what is the third? Unwinding of DNA helix. When the parental DNA is treated with the helicase enzyme, automatically what happens? The coiling will remove and they will form what? Unwinding. Okay, that is replication fork-like formation will take place and they will separate and there is no further coiling there. Okay, so after this, fourth step is formation of RNA primer. What is RNA primer here? RNA primer is a small segment here which is having 3' to 5' direction they will be having, and that is synthesized on DNA, the dependent on DNA dependent RNA. What is RNA primer? The RNA primer is a DNA dependent RNA. Okay, once the RNA primer is, so wherever the new synthesis taking place, there RNA primer will form. Especially that RNA primer start moving from 5' towards the 3'. Okay, so it is moving in this way and continue formation will take place on the first strand. On the second strand, it is not continue, but instead it is discontinue fragments. That fragments are called as Okazaki fragments. Okay, Okazaki fragment. Next, after RNA primer, now what happened to this RNA primer? They are going to form, just go on adding the, and also hydrogen bond formation also will take place, and go on adding the nucleotides and form continuous layer, that is copy. So RNA is with the help of RNA primer, the newly synthesized DNA has formed the first layer strand, that is a leading strand. Okay, next. After base pairing is added, and simultaneously, once the RNA primer also should be released with the help of RNA polymerase. Okay, with the help of RNA polymerase, we can remove RNA primers. And now elongation of the strand. The fragments, Okazaki fragments, is treated with the enzyme so that they will form the polydiester bond. Okay, hydrogen bond formation has taken place. Okay, now only thing was the which is present in the form of fragments, they have to convert them into elongated. They should be elongated by using the enzymes. Polymerase DNA, polymerase enzymes we are going to use. Next, after elongation of the strand, next is termination. Once here, as well as the fork formation, with the help of topoisomerase, the fork will separate and this will form as a new template, and there will be no coiling in this fork also. Okay, this is the last step is, once the newly synthesized DNA has taken place, sometimes A will be coded for the, A will be bounded with the double bond, as well as G will be bounded with the C, that is triple bond. So this bond formation will be taking place here, and once the hydrogen bond will be formed, and almost I told you during the nucleotide, again they have to convert themselves into monophosphate by pyrophosphatase enzyme. They will form this, and releases two inorganic phosphate and hydrogen bonds will be created there, and continuously because of the DNA polymerase proofreading. If instead of coding with A with T, instead of T, if it is coded with the U, then this will become wrong. So these type of mistakes are rectified by DNA 3 and DNA 1 also. They will not only rectify, they will not only identify the mistake, but also they will replace the correct nucleotides there. Okay, they will also replace also, especially DNA polymerase 1. Okay, so this is about the mechanism of replication. Go through. This is one of the very important question for 5 marks. Okay, next class we will start with the other topic.